Okay, I think I'll just start by welcoming everyone to this session of contributed talks. So just some procedural thing as before, you're used to this now, but please ask any questions in the Q&A tab. And please, since we have multiple speakers, prefix the question with the name of the speaker so that they're asked to the right person. And you can also, of course, upvote other questions that you're interested in. So with that, I'm happy to introduce our first speaker, Alejandro Reyes from the Novartis Institutes for Biomedical Research in Basel. And Alejandro will talk about how large-scale topological changes restrict malignant progression in colorectal cancer. So the screen is yours, Alejandro. I'm looking forward to your talk.
Thanks, Charlotte. Can I quickly get some feedback if you can see my slides?
Yes.
Perfect, thank you. So as a disclaimer, as Charlotte already mentioned, I'm employed at Novartis, but actually the work that I'm going to show today is work that I did as a postdoc with Rafael Heidari at the Dana-Farber Cancer Institute. What I'm showing here are two pathology slides. The left one is of normal colon tissue, and the right one is of colorectal cancer. And in the zoom-in regions, I'm zooming into the nuclei of these tissues. And in colon tissue, you can see that the nuclei tend to be small, roundish, and quite uniform. And however, in colorectal cancer, this nice small round shape tends to get lost, and you start seeing a lot of structures like this, and that are not the same between the different nuclei. They are enlarged, and they should lose their round shape. So a question that this is something that pathologists actually use to diagnose cancer, but this also raises the question of what are the molecular changes that occur in how the 3D genome is folded in the genome. And to answer this question, we have generated a large research of data consisting of a cohort of patients of 7 normal tissues, 26 colorectal cancers, and 6 cell lines for validation. And this data set consists of high-C data, high-ChIP data, DNA methylation data, ChIP-seq, and RNA-seq. What I'm showing here is the number of libraries that, for example, we have more than 50 libraries of high-C. And this, of course, shows the magnitude of this study, but it also shows the opportunities for reanalysis of this data set that I think in this kind of communities we really like, sort of like free data. As a small reminder of how we are measuring the 3D genome interactions, high-C data is a protocol in which if 2 regions in the genome that are not necessarily close in the 2D space are interacting in the 3D space, the protocol would cross-link DNA. It would cut this DNA with restriction enzymes, fill the ends with biotin, and then there will be a ligation step, a purification and shear down of the DNA, and a pull down of the biotin, such that the fragments that you end up are fragments of different regions of the genome that are closely interacting in the 3D space. If you subject this to high throughput sequencing data, you can then afterwards summarize your data in matrices like this, in which the genome is represented in the diagonal, and you can fill this matrix with the number of interactions that you see between each pair of regions. So a red bright color in this matrix would represent regions that, for example, this region interacting with this region, and so on. Thanks to these technologies, researchers have been able to discover that the genome is folded in a hierarchical structure consisting of chromatin loops, topologically associating domains, and chromatin compartments. So in this talk, I'm going to show you what are these, I'm going to describe what are these different structures, and I'm going to be telling you how each of these is disrupted in colorectal cancer. Enhancer promoter loops are regions of the genome that are far away in the linear distance, but they actually interact very closely in the 3D space. The most common type of these loops are enhancer promoter loops, in which distal regulatory elements, enhancers, bind to the promoter of genes to regulate their expression. When we call these enhancer promoter loops, or loops in general in our data set, we recovered around 15,000 loops. And something that was immediate from this data set was that the anchors of these loops were enriched for enhancers containing SNPs. And this data could be actually used to link candidate SNPs to candidate genes that these SNPs are associated with risk of colorectal cancer. So this data could be used to link distal genes to these SNPs. And here I'm showing 2 examples of the CR4 gene and the third gene, how these are linked to distal SNPs. Moreover, when we tested for differential loop between the tumors and the normals, we detected 571 loops that were stronger in the tumors and 248 that were weaker in the tumors. These gains or loss of loop strength were also associated to changes in gene expression, as you can see in this plot. Loops that were gained in the tumors were consistently associated with increased gene expression, where loops that were lost were associated with loss of expression in the tumors. This is an example in which this gene, PDC-B4, it's a tumor suppressor gene that is linked to an upstream enhancer through this loop that is significantly getting lost in the tumor samples. And when we went and looked back at TCGA data, we saw that indeed the expression of this gene is quite low in tumors compared to the normals. So all of this indicates that enhancer promoter loop changes contribute to oncogenic program rewiring. Next, we look at topologically associating domains. Topologically associating domains are consecutive regions of the genome that interact a lot with themselves and not with the neighboring regions. If you look at the Heisey matrix, if you zoom in the diagonal of this matrix, you would start seeing these triangular structures, which are precisely topologically associating domains. So this, for example, would be a region that interacts a lot with itself, but not with the neighboring tab and so on. You can see these sort of like triangular structures across the diagonal. When we looked at our cohort of data, cohort of samples, including the cell lines, we saw that the TAT structures tended to be very conserved, as shown here by the correlation coefficient. Whenever we saw a TAT boundary, a TAT in one of the samples, it tended to be conserved across all of the samples. This, however, there was an exception for a subset of tumors that are called the SIMP tumors, which stands for CpG island hypermethylator phenotype. What I'm showing here is the CpG island distribution, the CpG island methylation distribution of our normal samples and our tumor samples. In this set of CpG islands, the normal samples tend to be hypomethylated, as you can see here in the green boxes. However, the tumors, there seems to be a gradient of how methylated are the CpG islands. And there are two tumors that are really highly methylated, that have these islands really highly methylated. And these are our, we call these our SIMP tumors that we profile with Heisey data and other marks. The reason why this is important is because whenever a CpG island is hypermethylated, it inhibits the binding of proteins. For example, what I'm showing here is we divided the sites where the CTCF binding sites that were hypermethylated in the same tumors, and we also defined a set of non-hypermethylated CTCF sites. And then we compared the binding strength, the average binding strength in the normals, the non-SIMP tumors, and you can see that in the normal and the non-SIMP tumors, these regions don't change their binding a lot. However, in the SIMP tumors where these regions are hypermethylated, we also lose CTCF binding. The reason I'm telling you this is because CTCF sits at the boundaries of TAT structures. And actually, TAT structures are thought to be mediated by the binding, created by the binding of these proteins. So what we ask is what happens when we are losing these CTCF sites due to hypermethylation. And when we did this analysis, we measured the strength of this loop, which would be the interaction between two CTCF sites forming a TAT. For the sites that were not hypermethylated in the SIMP tumors, we didn't see a change in this loop strength. This is the average loop strength across samples. However, for regions that were hypermethylated in the SIMP tumors, we see that the average peak strength was really lost, meaning that the TAT structures were compromised. Despite having these differences and compromised TAT structures, we didn't see big gene expression changes that made us investigate further. However, it was perhaps more exciting what we saw for compartment structures. Compartment structures can be seen as water and oil. For example, if you have a subset of TATs that are highly active, that have active chromatin marks, they will tend to interact between each other and not with B-TATs that would be silent TATs. So these two regions were called A and B. For A would be the active DATs, B would be the active DATs. The way these are defined in the Hi-C matrices is by doing an eigenvector decomposition of this matrix, of a transformed version of these matrices, and the sign of the first eigenvector would give you the label of A or B compartments. In our analysis, when we tested for differences in this compartment structure between the tumors and our normals, we didn't see striking differences. However, the most striking differences were seen when we did some polymer modeling of the Hi-C data. Here, we team up with Ji Feng and a physicist from MIT, that what he does is he simulates chromosome structures, and then out of these simulated structures, based on an energy function, he then simulates a Hi-C map and asks how different is the simulated Hi-C map to the observed Hi-C map. Then he iterates between these, he iterates the parameter of his model until reaching a simulated Hi-C map that would converge to the observed experimental Hi-C map. When Yifeng did this for our data, here I'm showing two structures for two tumors, and you can see that B chromatin is nicely positioned at the periphery of the nucleus, while active chromatin is more positioned to the internal of the nucleus. However, for the tumors, this nice peripheral partition of chromatin compartments were lost, as you can see in these two samples. This is something that we could validate experimentally, and I'm showing you two microscopy images in which indeed heterochromatin is typically at the nuclear periphery, while in the tumors we would start seeing these perturbations of heterochromatin that get internalized in the nucleus. Also, another difference in compartment structure that we observed are highlighted here. So when we were just visualizing the eigenvector that is used to define the AB compartments, we would see these regions that tended to change from an A-like state in the normals to a B-like state in the tumors. And interestingly, when we overlay this with hypomethylated blocks, which are regions of the genome that tend to lose methylation in the tumors compared to the normals, we saw a very good overlap between these regions. Something that we also saw was that these hypomethylated blocks in which tumors would lose methylation compared to the normals were a lot of times overlapping very nicely with the topologically associated domains, meaning that this hypomethylation is actually restricted to TATs, and it's probably the whole TAT structures that get hypomethylated. This observation of this overlay with hypomethylation blocks is interesting because these have been typically associated with compartment B. And what we were seeing is that these also occurred in what would be called compartment A in the normal samples. When we looked at our chromatin marks, we saw that the hypomethylated blocks in compartment A were highly enriched for H3K27 remethylation mark. And this made us go, and this was especially strong for the tumor samples. This made us go back and look at the matrices, Hi-C matrices in more detail. And we saw that these regions, these hypomethylated A blocks, had a very particular interaction patterns. While B chromatin tended to interact with other B chromatins, the yellow marks here, and A chromatin tended to interact with other A regions. These hypomethylated A regions tended to have like intermediate interaction patterns between A and B, as shown in these three examples. Something that we also saw was that when we went back to the definition of the compartment calls, we could see that these points, hypomethylated A blocks, tended to separate in lower PC loadings of the eigenvector decomposition. The polymer models positioned these hypomethylated A blocks in the intermediate between A chromatin and B chromatin, as shown here. And this is something that we also validated using oligopainting, chromosome oligopainting. As you can see in this example, hypomethylated A blocks tended to have their self-interactions and tended to be between A chromatin and B chromatin. But these all suggested what we were actually seeing a novel compartment with intermediate interactions between A and B, intermediate gene activity and gene density. intermediate nuclear peripheral positioning, highly marked by H3K27 trimethylation, which is a mark for facultative heterochromatin, that were hypomethylated in tumors and were changing their interaction patterns towards a more B-like state in the tumors. These, of course, are very nice findings and highlight what are the changes that are changing in cancers compared to normals. But then we were asking, why are we seeing these changes? What do they mean biologically? And what I'm showing here is just a summary of what I just described, in which I see the y-axis, you see plotted the difference in PC1 between tumors and normals, and in the x-axis, you see the DNA methylation differences. And you see that the regions that get more hypermethylated, both in the A and the I compartment, this newly defined compartment, they tended to have more B-like states. Something that we observed was that in literature, it has been described that these regions are, the similar changes have been described for cells with accumulated cell divisions. And we actually went back and did a passage experiment in which we passaged WI38 cells and sample across different timelines, and we could recapitulate the same effect. That regions, this is comparing 2 time points, passage 40 with passage 16, we could recapitulate the same effect of DNA methylation losses linked to these differences in chromatin structure, compartment structure. Meaning that the changes that we were seeing in the tumors were due to accumulated cell divisions. Then we further, since there was this nice link between hypomethylation and the compartmentalization differences, we asked whether DNA methylation was likely to be causal. And to this, we treated HCT116 cells with a demethylating agent, 5-ASA, and we could recapitulate the same thing. Meaning that DNA methylation is likely to be causal of the 3D changes that we were observing. Now, we then ask, why are we seeing these changes? And since we have this nice link between hypomethylation and changes in compartment structure, we use the CGA data to associate hypomethylation as a surrogate of topological changes with changes in gene expression. Interestingly, what we see in these compartments that get hypomethylated were that a lot of genes were repressed, and these genes tended to be enriched for metastasis and processes like related to metastasis. And the genes that were induced, such as cancer germline antigens and ERF1 repeats, are things that are inducing immune responses. So all of these were consistent with these changes being tumor suppressive. We built a score based on these expression profiles, and we saw that the genes, the tumors that fail to repress these genes, were associated with worse prognosis. So with all this, we could conclude that what we were seeing is a mechanism that silences genes to restrict tumor progression, and the failure to repress this, to induce this mechanism, is associated to worse outcomes. With this, I would like to quickly jump to my conclusions, in which I can tell you what I told you before about enhancer promoter loops contributing to gene expression changes in the tumors, that are likely stable, except for zinc tumors where the boundaries are hypermethylated, and from compartment structures we see changes in associated hypomethylation that induce tumor suppressive gene expression programs. Something that I would like to mention is that these are the authors of all the packages that were used to analyze this data for this project. And you can see that there are a lot. And this is a reflection of how communities like this enable analysis like the one that I just presented in a very easy way. So in a way, all of these are contributors. If you happen to be close to where am I guess beer or coffee on me, as long as you are not all together coming at the same time. And with this, I would like to jump to my conclusion slide. And when I make acknowledgement slide, just acknowledging Sarah Johnson, which is experimentally does this project, and also the people that supervise this project, Bradley Bernstein, Martin Naye, and my postdoc supervisor, Rafael Irsari. And with this, I would be happy to take questions if there is any.
Thank you very much, Alejandro. This was very interesting and an impressive collection of data. So while people are thinking about their question, I see there is a raised hand, but please put your question in the Q&A so that we have everything collected there. So just while that is happening, I have one question which is maybe, so you have a lot of data, right? You showed how many samples you have and how much different types of data. And I was kind of wondering, do you still think that there is some additional modality of data that would actually help you draw further conclusions on what's actually happening here?
Yeah, definitely. So, I mean, this also brings me to one reflection. So, I think, for example, some of the chromatin marks that we sample, we can make some conclusions out of them. But I think it will also be there are marks playing roles in tumor progression, etc., that we didn't sample. So I think this could be added further to this data set. And also this brings me to the other part of, like, since this is a huge data set and we had limited time to analyze it and to get it out, there is a lot of opportunities for reanalysis. For example, there is the whole question of how copy number variants are contributing to the changes in topology that we didn't assess here. We just controlled that the observations and conclusions that we made were not due to copy number variants, but this is something that could be addressed further. The same with translocations. So I think this is a very good resource for developing projects further.
Yeah, I agree. So are you planning to make the data into an experiment hub package or so?
Probably yes, that's a good idea. I should do that. I have at the moment all the process data is on Geo, and I have a package on GitHub that is still in progress, in which I show how to reproduce each of these figures from the process data. This still needs a little bit of work, but I think it would also make sense to make it available through an experiment hub.
Great. So we have a couple of questions. First, there are a couple of maybe less serious questions. People wondering if they can steal your bioconductor contributor slides for their talk.
Yes, I can also post the code that I used on GitHub.
That was another question, or the code. So, so there's a question from Davide who's wondering, actually, yes, this is a great multimodal resource. Is it available in bioconductor? And I guess you kind of answered that it will happen, hopefully.
Yes. At the moment, it is available on Geo, though. All the process data is there. The raw data is not on Geo because of safety concerns, like ethics concerns. We need to still do the paperwork, finish the paperwork, and finish the uploading to dbGaP.
Yep. So then there's a question from Aideen who's wondering, can you comment on the role of ERV? How did you define ERV genes? Did you stratify by viral-induced tumors?
Here we used a notation from, it's a very good question. Here it's a simplified version of, we just used annotations from repeat masker. And we just overlay, we just consider the reads that would overlap with this, the unique parts of the ERV1 repeats.
There are no more questions right now. So if you have other questions for Alejandro, just reach out during the conference and ask them later. So with that, thank you very much. This was a great talk. So our next speaker is Elias Haydari from ETH and the University of Zurich. And he will talk about the method to leverage gene networks for single-cell annotations. So we can see your slides. So I'm looking forward to your talk, and the screen is yours, Elias.
I'm Elias Haydar. I'm a master's student at ETH in Zurich. I'm also a research assistant in the Robinson Lab at the University of Zurich, working on single-cell data. And today I'm going to present a scGCN for leveraging gene networks for single-cell annotation in collaboration with Shahan Shakarfurush and Lale Haqverdi. So cell annotation is a pivotal task in single-cell research nowadays, and quite a few methods have been put forward for annotating single cells. However, almost all of the contemporary methods treat genes as independent features or independent groups of features. In contrast, here we want to propose how to incorporate the global gene interactome as an inductive bias to facilitate a more reliable and robust annotation. So our first motivation is cross data set transfer learning. And as the name suggests, in transfer learning, we want to transfer our knowledge that we've gained on one environment to another environment. In other terms, we want to generalize our models to unseen environments in a robust and accurate manner. And this is involved in many applications, as we've seen in the keynote speeches yesterday and also in the first day. This is involved in many applications, specifically when we lack reference data sets, where the unannotated data set in hand is acquired on, for example, new studies, new organs, new species, new states, or new modalities. So we want to look for the common features between the observed and the unobserved environments. One invariant feature is the gene interaction network, in which we know which genes specifically are connected to what other genes. And this can be also perceived as the backbone of the gene regulatory network. The other motivation is to define a better single cell metric for the single cell space. So let's take a look at this hypothetical tiny example, where each row represents a cell and each column represents a gene. And for now, for simplification, just consider on or off states for each gene. And if we compute the Euclidean distance as we do in reference-based annotations or clustering-based annotation, we will get such a topology where A, B, and C are equally far apart. While if we project the activations on the gene regulatory network, we will see a different topology or different geometry where B is an intermediate cell between A and C. So how do we do that? How do we incorporate the gene network? The first step is to reconstruct the gene network. So this can be done in many ways. One way is to use prior knowledge, KIC-like databases, which are publicly available. But one other way to do that is to reconstruct the gene network based on co-expression. And I'm really lucky that Monica today set the stage for the undirected graphical models in which we find the conditional independence and conditional dependence, and this gives us a robust way to connect the objects in hand. And here specifically, so this is the graph the network we reconstructed based on zebrafish and Brio dataset. So also for future reference, each node here represents a gene, and the brightness of the node represents the level of activation, whatever it is, whatever the measurement is. The brighter, the more active the gene is. And we connect two genes if we connect two genes if they are statistically relevant in conditional dependence terms. I won't go into detail here, but in the end, what the graphical models, these graphical models gives us a sparse and also a modular graph when applied to single cell data, and this is very favorable for the sake of computation, also for interpretation, and this is in line with our knowledge of biological processes, that groups of genes are active or inactive in a specific biological process. So to wrap up this step, so we reconstruct this based on our first environment, based on our observed data set, and we use the same network. We feed in the same structure to the model for annotating the unobserved data set. So this is our inductive bias, which is the same across all of the cells. The only thing that is different is the activations over this graph. So now that we have this graph, how do we do the inference based on the activations? So let's take a look at differentiation processes in nature. So what we have in differentiation processes, we know that some genes interact with each other, and then some processes happen, and in the end, we will have some some specific cell lineages and differentiation processes. So we want to somehow simulate such processes. We want to diffuse the information over our graph so in a smart way, so that we will be able in the end to use that diffuse information for our annotation. So how do we do this information diffusion, or so-called message passing, or information propagation over these graphs? We use deep neural networks, because they are able to, in my terms, simulate any complex diffusion process or propagation process. And specifically, we use graph convolutional neural network, which are applicable specifically to graphs. And there is a whole body of literature in the deep learning field nowadays on that. I won't go again into details, but this is to give you an intuition about what's happening behind the scene. So when we talk about diffusion information or message passing, we mean, so if you consider one node, we want to aggregate information from the neighbors. We can go further and aggregate information from neighbors of neighbors, and this can go on to k steps, where we aggregate the information from a certain depth of the neighborhood. We call this k the locality, which means the local sphere that we put around each node to aggregate information. And each of these aggregations are weighted by a certain filter, and we call them Chebnet filters. Again, I don't want to go into detail, but we can apply these filters many times, let's say n times, and then in the end we will be able to simulate the very complex diffusion. Of course, how we do that, how we aggregate data, the weights are learnable parameters, and we learn them, and this is specifically our goal in annotation. So if we take a look at our reconstructed graph, and this is the case for most of the data sets we've worked on, we see that the graph structure is somehow heterogeneous in terms of density of nodes. And therefore, to capture this heterogeneity, we have to use different spheres with different radius. So in order to do that, we use a specific graph convolutional neural network, which is called Inception GCN. We apply these filters multiple times for each node, and this is a common structure of each deep neural network, which we do computations layer by layer, and in the end we do our inference, which is here annotation or classification. So just to mention that, so we start from the raw gene network that we reconstructed. The only thing that is different for each cell is the activations over that. We apply these layers which diffuse the information based on the input activations, and in the end we will have something, again the same structure, but with a complete different distribution of activation over the graph. And we put it as an input to a dense layer, again a deep learning term, and then in the end we will have our prediction. So here, for example, it can be cell type or any other annotation of interest. So to wrap up the design part, so we start from the single cell omics matrix, whatever it is. It can be RNA-seq or anything else. We should do quality control, of course, and then normalize and scale based on the input range. Then we have to subset genes and cells to make this computationally feasible. Then we reconstruct our gene network. Once we reconstructed our gene network, we have to design our graph convolutional neural network based on the topology of the gene network, and then in the end, we will have our predictions, and then we can validate by comparing to the ground truth. So this is just one data set that I will present in this talk, but this is a very cool one. So this is a public single-cell RNA-seq data set. Under this name, we downloaded it from the GEO. So it contains data from 7 lung cancer patients. From each of them, we have 2 tumor samples, lung tumor samples, and we have 1 blood sample. We also have 2 healthy mice and 2 cancer mice, again sampled from their lung. And our task here is to do cell type annotation. So this data set is really cool because we can define multiple transfer learning tasks here, for example, cross-organ, cross-state, and a very complex task, including cross-organ, cross-state, and cross-species at the same time. So this is a very hard task. So just for completeness, we mapped the homolog genes from the MGA portal. We also did a very naive normalization with the SCRAN, and also selected 200 highly variable genes with the SCRAN. So this is what happens in our neural network. So let's first take a look at the raw data. So if we project the raw data to our graph, so let's first tell you what are these graphs showing. So each of these nodes represents a gene, and the brightness is proportional to the average of activation over the whole set of cells, which lie in the specific group that I mentioned here. So, for example, here for B cells, we take the average of activation for the genes which are present here, and then we color the graph based on the activations. This is for B cell, and this is for natural killer cells. So if let's take a closer look to the graph here. So if we use Euclidean distance here, for example, we will have some confounding results, some asparous results, because this community here is shared between natural killers and B cells. And this community can make them close together. This can be a confounding factor that can result in a vague classification. So let's now see what happens in our graph neural network. So the first layer, as I mentioned, so it diffuses information from the from the more activated parts of the graph to the less activated parts of the graph. So if you look at the first hidden layer, so the activation is all over the place. But if you look at the final hidden layer, so here the hidden layer 2, we will see that again the activations are accumulated on some specific parts of the graph, and the activations are accumulated far apart in projects on the graph. So, for example, for B cells, we have an activated cluster of nodes here, while for the natural killer cells, we have an activated community over here. And now we will be able to distinguish these 2 cell types better. So this is just to give you an intuition what happens in the information diffusion process with our neural network. And these are our validation results for each of the tasks. So we compared our model to a baseline model, which is fully connected neural network, and so which doesn't use the network information. And I want to draw your attention to a couple of things here. So if you look at this column, which shows the test accuracy, we did a better job in all of the tasks in comparison to the fully connected model. Well, if you look at the train accuracy, the fully connected model outperforms our model in all tasks, and so this has a conclusion that the difference between train accuracy and test accuracy is much lower in our case, and this is a favorable feature of a classifier when we are doing transfer learning, because if we have a specific train accuracy, we can be almost sure that test accuracy is not far away. While if we use fully connected model, it's not the case. But one other thing to be fair to mention is the average running time, which if we look at our model, it's much higher. And this is because of the whole complex computations that our model performs. And so maybe there is room for improvement there, but we cannot go further from some specific point. And this is how we implemented this. So there are multiple decisions to make: how many genes I want to work with, how I do pre-processing, how many cell types I pick, how we reconstruct my gene regulatory network based on prior knowledge, based on which graphical model, and so on. So in order to be able to benchmark all these hyperparameter constellations, we implemented this as an R package, which is still dirty, but it's still ongoing, and we are trying to improve it. And this is here. You can see the code there. One other problem that we have, if you want to put everything together and do an end-to-end optimization and end-to-end benchmarking, we have a module, our classifier, or if we want to add other classifiers, we have to deal with Python, and this makes things a little bit complicated because there will be some dependencies that makes this difficult to maybe in future submit it to Bioconductor. So we have to deal with these difficulties, but it's ongoing, and I guess we will be able to make a big progress in future months. So in the end, I want to wrap up things with mentioning that the scGCN sheds light on leveraging gene interactions networks for single cell annotation, and it's applicable to any categorical annotation tasks, not necessarily cell type annotation. In particular, scGCN aims at using such an inductive bias to improve transferability of annotation models. And in training, scGCN is now quite slow due to the complex computation it performs. So maybe there is room for computational optimization. And scGCN utils, our R package, is designed for benchmarking gene network-based annotations. You can replace our modules with any other module with the same purpose. And there is an open question still, which is fundamental limits of graph convolutional networks in single cell annotation. With that, I want to thank you all for listening. Also, thanks to the chairs and the organizers of the EuroBioC. I also want to thank my collaborators, Shayan and Lale, and also specifically Huber Group, which this project initiated in 2018. Specifically, thanks to Simone and Wolfgang. Also, our funding resource sharing Stiftung and the University of Zurich and my group, the Robinson Group, and ETH Zurich for providing me with their GPU servers. And I will be open to questions if there is any, also on the platform in future hours.
Thank you very much, Elyas. This was great. We have a fair number of questions. So I will start with Michael, who's wondering, how would your model compare to a simpler label transfer approach, like the one in single-R based on correlations?
Yeah, so yeah, this is a very good question. So so in the beginning, we wanted to do simple single cell annotation, like cell type annotation. But yeah, so all of these methods, so we compared to some of them, and many of them do not do a good job when we have complex transfer learning tasks. So because, as I mentioned, they do not use a specific invariant feature. And the reference, so one thing is that what about the marker-based or the reference-based annotations? And that's correct. But again, when we have a transfer learning task, which we cannot transfer the knowledge, for example, of the same, we cannot use the same markers, the same reference cells, then there would be some problem. But when we use these interactions, which are invariant, we will be able to transfer our model. About correlation-based methods, so the correlation-based methods, they also include the transitive edges, which we drop because of the conditional independence phenomena.
So you're kind of including different types of models as well in your benchmarking, let's say. Okay, then there is a question from Johan, who is asking how much the results of your approach depend on the initial input GRN. So he assumes that this step is crucial, also difficult, and how do you get these GRNs for your example?
Can you still see my screen?
Yes. Yes.
Yeah, so it's really a relevant question, and it's a very good question because it's really the accuracy of the model really depends on the quality of the gene network. Sorry, and if the true gene network underlying the first data set is consistent with the second data set, then the model will be transferable very well. But if that's not the case, then we will be in problem. So it plays a very important role in our model, and we do again some optimization and some scoring of how consistent are the graphs constructed on the first data set and the second data set, and we do some decisions there to make it the most coherent possible.
Okay. So I'll take one last question. So there are a couple of pointers to the Basilisk package, which may help you to specifically make the R to Python bridge for bioconductor. But then there is a question from Luke saying that 200 is a very small number of genes, right? So how can you be confident that your gene network actually is a good one when most of the genes are not used?
Yeah, so it's a challenge when we go to further, our computationally, we can go to about 1,000 genes or even more, but it was a challenge to see if we restrict ourselves to very low number of genes, then what would happen. And so this is also in line with our our second motivation, which is defining a better single cell metric. So we know that there are some, so when we use the Euclidean metric, there are some, like for example, when we look at the UMAPs, we say that from RNA-seq itself, we cannot decouple two subpopulations. But we hope that by using this inductive bias, then we will be able to better deconvolve or separate the subpopulations.
Okay. Okay, so in the interest of time, I think we'll round up here. Were a couple of questions left. So if your question was not answered, please just I encourage you to reach out to the speakers in the platform during the conference. So thank you very much to both of the speakers of these sessions for great talks, and thanks to all the people who are listening.